Hello, good afternoon. I'm Eloy Lozoya. I will be presenting my research project titled Mesenchymal Stem Cells Transfected with Osteogenic and Angiogenic Inducing MicroRNAs for Bone Tissue Engineering. To get into our introduction, it's important to state that bone is a very important organ in our body. It supports and protects our other organs. It allows for movement of the body. It's important in producing progenitor cells such as red blood cells and white blood cells, but it's also vital in maintaining mineral homeostasis such as calcium blood levels. As we know, bone can suffer detrimental injury to it, such as fractures or have bone defects to it. Sometimes these fractures can regenerate by themselves. However, there are some instances where medical intervention is needed to aid the healing process. The medical intervention used is bone grafts. However, it comes with many complications, such as blood loss, risk of infection, and sometimes the need for multiple surgeries. An alternative to the use of these invasive bone grafts is something called bone tissue engineering. It is comprised of biomaterials, cells, and bioactive factors either by themselves or in combination to regenerate a functional bone by using the body's own cellular processes. And this project will be focusing on the bioactive factors, which in this case are the microRNAs. Our target here is to promote osteogenesis, which is bone formation. However, we also focus on angiogenesis, which is the proper vascularization or blood vessel formation to allow for these bone cells to have proper nutrients and the ideal conditions to proliferate and grow to promote a functional bone again. Traditionally, the bioactive factors that have been used to differentiate these stem cells into bone cells have been proteins. However, they're very difficult to manufacture and they're expensive. The alternative bioactive factor that we introduce here are microRNAs as the alternative bioactive factor. MicroRNAs have many benefits, such as they can regulate multiple genes and pathways, but the most important is that they regulate in vivo production of proteins to keep these proteins in their most biologically active form. We have done research and found that microRNA 26A has been shown to induce bone regeneration by influencing both osteogenic and angiogenic coupling. So it's a promising alternative approach for proper bone formation and bone tissue engineering. The hypothesis for this project is that the 326A microRNA variants will vary in effectiveness in inducing osteogenesis or angiogenesis in our transfected human mesenchymal stem cells. The objective, however, is to evaluate which of the 326A microRNA variants is most effective in inducing osteogenesis or angiogenesis in our transfected human mesenchymal stem cells. Moving to our methods, we found the 3 osteogenic and angiogenic inducing microRNA 26A variants through literature research. Once we found the microRNAs that we're going to use for this project, we cultured our human mesenchymal stem cells in complete cell media, which is basically alpha minimum essential media, supplemented with FBS, penicillin, and L-glutamine. After the cells were cultured and left to grow, we transfected our cells with microRNA using lipofectomy to measure mRNA levels of our target genes. 7 days after transfection, we did RT-qPCR to measure the mRNA levels. For our protein levels, we did immunoblotting after 7 days as well. And for our calcium deposition, we performed alizarin red staining to measure the amount of mineral deposition in these cells at day 7 and day 14. Something different that we did for calcium deposition is that we cultured our transfected human mesenchymal stem cells in osteogenic media rather than complete cell media. Moving to our results, seeing our RT-qPCR data that measured the mRNA expression of our target genes for RUNX2, which is an important osteogenic marker, we saw that the human mesenchymal stem cells that were transfected with all 326A microRNA variants expressed significantly higher levels of RUNX2 mRNA compared to our negative control. Moving on to ALPA, which is another important osteogenic marker, we saw that the human mesenchymal stem cells that were transfected with the microRNAs 26A13P and 2,3-P express significantly higher mRNA levels of LPL compared to our negative control. Moving on to VEGF, which is an important angiogenic marker, we saw that the human mesenchymal stem cells that were transfected with all three 26A microRNA variants expressed significantly higher levels of VEGF mRNA compared to the negative control. Moving on to VGLAB, an important osteogenic marker, we saw that the human mesenchymal stem cells that were transfected with the microRNAs 26A13P and 23P expressed significantly higher mRNA levels of VGLAB compared to our negative control. Moving on to our immunoblotting data. Looking at RUNX2, the osteogenic marker, we saw that cells that all the human mesenchymal stem cells that were transfected with their microRNA 26A variants express higher protein levels compared to our negative control. Looking at VEGF, we saw that the human mesenchymal stem cells that were transfected with the microRNA 26A13P express higher protein levels of VEGF compared to our negative control. Looking at angiopoietin, an important angiogenic marker, we saw that the human mesenchymal stem cells that were transfected with all three microRNA 26A variants express higher protein levels of angiopoietin compared to our negative control. Moving on to our calcium deposition data, we saw that when human mesenchymal stem cells were cultured in complete cell media, there was no alizarin redis staining, which is why we decided to culture or transfected human mesenchymal stem cells in what is called osteogenic media. At day 7, the human mesenchymal stem cells that were transfected with the microRNAs 26A5P and 23P and were cultured in osteogenic media showed darker staining compared to our negative control and cells transfected with microRNA 26A13P. At day 14, we saw that the human mesenchymal stem cells that were transfected with all three 26A microRNA variants and cultured in osteogenic media had darker staining compared to our negative control. Looking at our conclusion, the data suggests that the microRNA 26A variants can promote osteogenesis and angiogenesis in human mesenchymal stem cells that were transfected by upregulating our target biomarkers associated with the osteogenic and angiogenic pathways. When we compared three 26A microRNA variants, we saw that microRNAs 26A13P and 23P show higher effectiveness in upregulating our target genes and markers compared to our microRNA 26A5P. When we look at our calcium assay data, it is shown that osteogenic media might be necessary to allow for calcium deposition in these cells. Our future work consists of repeating immunoblotting to obtain data for our protein marker osteocalcin, which is a protein transcribed from our gene VGLAB. And also we want to repeat the immunoblotting rate just to confirm our data results of the immunoblotting. Something very important that we want to do in the future is to repeat immunoblotting and RT-qPCR. However, when these human mesenchymal stem cells are transfected and cultured in osteogenic media, rather than complete cell media, which was what was performed in the study. I just want to acknowledge the College of Health Professions Research Fellows Program for providing supplies and the assistance for this research project. I appreciate your time and thank you.